Hello and welcome to your Ocean Pollution Lab. So, in part one, we're going to be doing a little bit more of a hands on lab. We're going to be doing some more kitchen chemistry with these materials right here, all of which you'll probably find in your kitchen already. And we are going to simulate an oil spill using some cooking oil. Um, and what do we do after there's an oil spill? We try to clean it up, and that's exactly what we're going to be doing in this lab. So we're going to compare our methods for cleaning with some of the methods they used for that 2010 Deepwater Horizon oil spill, which was the largest spill in the history of the world. So this part is going to be a hands-on activity. And then when we get to part two, we are going to pretend that there was an oil spill in the Columbia River estuary, and we're also going to pretend that you are in charge with leading a team to clean up that spill. And so, to strategically clean up that oil that is dumping into the estuary, you're going to use something called an environmental sensitivity index map. And basically, what this map does, it looks like this. It has different colors representing different habitats of different organisms, and you have different colors representing how sensitive different shorelines are to an actual oil spill. Some of them are more resilient, some of them are more sensitive, and they are damaged more. Um, Now, just to make sure we're all on the same page of where this map is located, let's zoom into the Columbia River estuary. Remember, an estuary is where that fresh water from the river is meeting the salt water from the ocean. And the map covers this area right in here. So a lot of the focus is going to be on this Baker Bay. Okay, so some parts of this map isn't as intuitive as it looks. You have a lot of overlapping data. So we have a whole nother PDF serves as a legend. Um, so there's two different types of data projected on here: the sensitivity of shoreline habitats and biological resources, basically the critters that live there. And these are going to be kind of overlapping, so you want to be able to tell them apart. So, shoreline habitats, these are going to be the solid colors. And what this legend doesn't describe well is that these are um, in order of sensitivity. So, these up at the top, these low numbers with the cool colors, purples, blues, these are the locations or the types of shorelines that are more resilient to an oil spill. They're not as damaged. But then as you get down to these red colors and these high numbers, 9 and 10s, uh, these are shorelines that are much more sensitive and can be more damaged by an oil spill. Okay, and then we have the different types of critters and organisms living in that area, and these are the hatcher marks that represent their habitat. And so if we look at the map, one part that gets a little bit confusing is that some of this data can overlap. For example, here we have the habitat for this crab indicated by the hatch marks. And then we also have this tidal area where there's this large splotch of color, and this is indicating the sensitivity of this shoreline right here. So both types of data on top of each other. Okay, you'll want to study this legend and also read this section because this tells you how to use the map. It also gives you the information that you need to answer some of the questions. Okay, finally, part three does not have to do with oil spills, but rather bioaccumulation and biomagnification. So there's a good description of these two in your textbook. Be sure that you are reading those and then regurgitating that information in your own words when you put the definition. But then you are actually going to be doing some calculations to see how biomagnification works, especially as you go up trophic levels. And so the first one is done for you. You can see the example. What you're looking at is the amount of a chemical called mercury chloride, which is a contaminant. It's a pollutant that interferes with a lot of the functions of the organisms, in this case in an estuary. A lot of estuary talk in this lab. Um, but a hypothetical situation that there is 0.04 parts per million of this mercury chloride in the estuary mud and detritus. This is a definition you should look up. That's a fun one. And then we have grass that is consuming some of those nutrients, and it is in turn consuming some of that mercury chloride. So how do we figure out how much the grass is getting? Well, you look how much is in the mud and detritus, and then you look how much of that mud and detritus that the grass is consuming, and you multiply them together. So here it's 0.04 parts per million times 10 units, and that is telling us that 0.4 parts per million is being transferred to that marsh grass. Okay, and as you go through the rest of these questions, you're going to build on the answers that you had in the previous questions. And then this last one, D, is the real kicker. Now you have a type of bird, songbirds, and they eat a variety of things. So what you're going to have to do is split up this 100 units between its different types of food that it likes to eat. And then you're going to have to do 4 separate problems based on your answers to A, B, and C. And then you add them up. If you are having trouble with this section or you want to run an answer by me, be sure to reach out. I'm happy to help. And then you have just one follow-up question there. Okay, let me know if you have any questions with the lab. Reach out to me and I am happy to help you out.